well, welcome today. I'm Ashley Capron. I'm the Director of Research and Science Operations at the Utah Clinical and Translational Science Institute. And I'm joined today by two leads of our Cellular Translational Research Core, or CTRC for short. They're here to present on one of the many facets of work that they do in this core. Our presenter today will be Dr. Colin McGuire. He's the managing director of this core, and he's joined by Alex Shligovitov, who is an associate professor of neurobiology and the faculty director of this core. So thank you for joining us today, and I'm going to turn this over to Colin.
All right, thank you for your kind introductions, Ashley. Let me just get this started here and share. Let's see. All right, can everybody see that? It's good?
Yes,
all right. Yeah, so thanks again for the introduction. Today I'm going to talk about innovations in our stem cell side of research work. I first joined the CTRC in 2015, and Alex joined us this January. And so this is a really good time for us to proceed in this work. So first I'll start by an overview of some of the services that we provide, and then I will give a few examples of some of the collaborations and some of the new innovations that we've incorporated into our workflow. So our main mission is to help clinical researchers make discoveries. And many investigators are looking at disease mechanisms, and we help process many of these biological materials, whether it's cells, tissues, or cell derivatives. And so we use this work for translational research. And we have three main areas that we support. The first is our DNA extraction facility. And this is a molecular biology facility, and we pretty much specialize in two areas. The first is extracting nucleic acid, and also we specialize in culturing primary cell lines. And also our main area, which we started, so back in 2015, we did not have a stem cell facility at the University of Utah. And because this lab had historically over 20 years experience processing human blood samples, it was thought that this would be a good fit for our core to also start the stem cell facility. And so we do three things. We can make induced pluripotent stem cells, we can genome edit these stem cells, and we can perform differentiation studies. Also, we house a biorepository, which holds a variety of different human cell samples and cell types. So just quickly with our DNA extraction facility, so you have a good understanding of what we do in our core. This is just a snapshot of samples collected during this past year. We have extracted over 6,000 DNA samples, nearly 500 RNA samples, and we have isolated PBMCs from whole blood and serum and plasma. Most of the serum and plasma and PBMC work was done with this large study called RAVEN through the CDC, where they were testing a new vaccine. And this past spring, we sent this entire collection of samples that we collected in bulk to the CDC for further analysis. So over the years, we've had several good collaborations, and one of them I'd like to note is with Dr. Sulji Lee during the pandemic. A lot of labs in California kind of slowed down or stopped, and so she contacted me and said, hey, are you guys still open? We have lots of very valuable samples that I would like to store. And I said, sure, send them our way. That work started by we started just storing these samples, but eventually we started processing them, and then it led to a paper that was published this last July, looking at factors that individuals that are asymptomatic with COVID, and that led to a Nature paper. So that was a very fruitful collaboration during the past 2 years. So as I mentioned, with all of our sample collecting, the last area is our biorepository services. And in 2015, we had very many old freezers that were mechanically failing. So when I got involved, one of my missions was one of my goals was really to modernize our biorepository and kind of transition away from all of these old freezers to a newer technology, which is liquid nitrogen. And in our freezers now, we have many thousands of different sample types, over 100,000 cell lines derived from 63,000 patients. All of this data we have been migrating into a LIMS system called Open Specimen. And as you can see, our footprint here on campus is pretty broad. There's 21 departments that use our core and over 60 investigators. And one interesting note during the pandemic is that we became a shipping hub. More than ever, lots of investigators and lots of larger collaborations, they need samples to go all over the country. And so, as you can see, we've in this past year, we have made shipments out to over 37 different institutions. So now I'd like to, this brings us to the stem cell portion of this talk, and I'd like to jump into that. And so really what happened, this started back in 2006, and that was the discovery of induced pluripotency. Before that, it wasn't even believed that once a cell differentiated, you could never return it to a stem cell state. But Yamanaka identified 4 factors that if you added them to cells, this controlled the stem cell state. For his work with Dr. John Gordon, he won the Nobel Prize for Medicine in 2012. And this opened up a whole new field that didn't exist. So just so you can get an understanding of, a basic understanding of the process of making stem cells, this is, you can take any cell from the human body if it is proliferating, and in most cases, it's most popular for skin fibroblasts to be taken, but subsequently, blood cells were also shown that you could reprogram them. You add these 4 Yamanaka factors into these cells, and then they return to a pluripotent state, and that process is called reprogramming. Once you have stem cells at that point, then you can differentiate them into any cell type of interest. And so probably from 2010 or earlier all the way to 2020 and beyond, lots of work was trying to find protocols that you could differentiate these cells into a specific cell type to study. And so this is kind of the general workflow, and we have like many labs, stem cell labs, we incorporated this workflow into our core. And so what happens here at the University of Utah is an investigator that's interested in studying a particular disease, they will give us either skin fibroblasts or whole blood. We will then take those samples, and in the case of human blood, we will isolate these cells, the blood cells inside them. And then we grow them in some special hematopoietic stem cell media for about a week to make them happy. And then we add those Yamanaka factors, OXOX, KLF4, and c-MYC. And then we use a virus-based method that just it doesn't change the DNA. It just goes into the cytoplasm. it makes these factors, and then those factors translocate into the nucleus, and then they reprogram the cells. That process takes about 3 to 4 weeks. You start to see these little colonies form, and then we pick them manually, and then we start to propagate them. After about another 3 or 4 weeks, then we perform some quality control. We look if there's any bacterial or mycoplasma contamination. We look and see if these cells are expressing the proper stem cell factors and remain in an undifferentiated state. We do some genetic fingerprinting to know where these samples came from. And then we also karyotype them, because the process of doing reprogramming is very harsh on the cells. And sometimes in about 10% of the samples, you'll get chromosomal changes that you don't want. So once you identify a line that has a normal karyotype that is identical to the patient that it was derived from, then they're ready to study. And so today we have generated over nearly 2,500 iPSC lines from over 180 patients. And this includes inherited diseases and acquired diseases, and also healthy controls. Once we have these stem cells, then we can also perform differentiation studies, and we can make very specific cell lines, running them through protocols. We can make cardiomyocytes, we can make neurons, and then we can also make 3D organoids. And in this case, we made retinal organoids a few years back for an investigator. And so currently we are working on new cardiac organoids. They make a cardiac structure, and then they beat, and so it kind of replicates heart development. And I'm also making some endothelial cells for investigators to study as well. So the process of making these iPSC lines and how they're used in translational medicine is, the purpose is really, since humans, in many cases, we cannot study like the biological mechanisms directly in the human body. So the purpose of this process is to make iPSC models to study human disease. And so in this diagram here, you can see that you take patient samples and you reprogram them back to a stem cell state with isogenic controls, and then you can genetically manipulate them as well, turn them into cardiomyocytes or whatever the disease happens to be, and then study them. Then identify targets, which can then be used in personalized medicine or in understanding disease mechanisms in general. So really, the fast part of this that's really getting developed right now is the CRISPR-Cas9 technology that is allowing investigators to specifically change gene sequencing and to understand what happens as a functional consequence of that change. This work was very, very, it was also a huge, major scientific breakthrough. CRISPR was discovered in 1993, and then it took nearly 20 years to develop it to the point where you could make CRISPR-Cas9 tools to be used in human cell lines. But for this work, Emmanuelle Carpentier and Jennifer Doudna, they shared the Nobel Prize in 2020 for the work that they did in developing these tools. So this was so groundbreaking, and this is a very active area, very fast-changing area of stem cell research right now. Like many labs, we have incorporated this technology into our pipeline. This overall process takes about 4 to 5 months to complete. Here at the University of Utah, we have a great set of cores that we collaborate with. So the first step is we collaborate with Crystal Davey. She works in the mutagenesis core, and she does, if an investigator comes to her, she will design a CRISPR, and then it will get made, and then that CRISPR gets dropped off at our core. At step 3 over here, you can see that we then take these guide RNAs and Cas9, and we electroporate them inside the cells of our stem cells. Then when it's inside the cells, it cuts the DNA at very specific targets. Then you get step 4, you get a pool of iPSCs that are either cut or non-cut or partially cut. There's lots of different combinations that can happen. Then we performed this assay called TIDE assay, and it looks at the overall cutting efficiency of these cells inside that pool. And so once it identifies, usually if it's like 30+% cutting efficiency, then our lab will know, okay, it did cut, let's continue with the work. So then once we have that pool, at step number 6, we will flow sort these cells that are cut and non-cut onto a plate in individual wells. Some of those cells will expand and grow, and then we expand them, and then we perform qPCR to see if there's actual genetic change at that location. Once we identify those clones that are actually manipulated, then we can expand them and hand them back to the investigator so they can do their functional studies. So this whole process, as I mentioned, is a long process, many parts. I think we collaborate with at least 4 different cores to get this work done. But it is a very important part and kind of definitely a new innovation and future innovations, which I hope will make this process even faster than it is right now. So next, I would like to kind of give you just a few examples of collaborations that we have done in our core over the past few years. So the first one I'd like to bring attention to is Dr. Jun Yang. She works in the Moran Eye Center, and she studies Usher syndrome. And this disease it's a leading cause of hearing and vision loss in the US, and it affects over 15,000 people. And it can happen in both adults and in children. And it's believed that 85% of these cases have mutations in this gene called USH2A. So Dr. Jun Yang approached us, and she said, hey, I have these patients that we'd like to study, and could you make some induced pluripotent stem cells for us? And I said, yes, we can do that for you. So she delivered whole blood from these patients from the clinic, and it was 2 affected individuals and their siblings as controls. So over the course of several months, we were able to create these induced pluripotent stem cell lines. And this is just an image of our quality control showing that these cell lines express OCT, NANOG, and TROP160. These are markers for undifferentiated cell state, showing that we actually have stem cells. Next, we gave these 4 stem cells lines back to Dr. Jun Yang, and she then did some further characterizations on them. This is a relatively large gene, but there's a deletion here at 299. You can see 2,299. And they also sequenced both sibling controls and the affected individuals, showing that their mutation is inside of these samples, so it got retained. And then she just shows a general pipeline of what we did during the characterization process. So at this point, she had some data that she wanted to publish, but she had a collaborator at the University of Miami that already was working on a similar project. And they were interested in taking these cell lines and then correcting them back to the wild type state. So we shipped these cell lines to University of Miami, and they did some sequencing, confirming that there was a mutation in these cell lines in the affected individuals. And then they performed a CRISPR experiment through homology-directed repair, where the CRISPR makes a cut at a very specific location, and then they insert an oligo with the corrected sequence into that site. And then you can see down here in clone 16 and 27, they found 2 clones that had specific that were corrected back to the wild type state. And so they were very excited about this, and this led to a publication in this journal called Genes, and that came out in, I think, 2021. And so we were really happy that this work kind of came full circle from start to finish, and we were involved in this project. So the next example I'd like to share is with another principal investigator named Dipayan Chowdhury, and he works at the CVRTI, which is the Cardiovascular Research Center here at the University of Utah. And he's interested in studying the role of mitochondrial dysfunction in cardiomyopathies. And so you can see here from this image, he's studying calcium unipolar channel activity. And usually his hypothesis is when there is a mutation in this channel, it affects the channel activity and increases the flow of calcium into the mitochondria, which creates eventually dysfunction. So Dipayan was, most of the way through this research, but the reviewers really wanted him to have not only some animal models and like fruit fly models of this channel, but also a human example. So he approached me and said, hey, I can get these fibroblasts from this infant that has a severe fatal lactic acidosis. And then what the plan would be is for us to make the line and then differentiate the cells in the stem cells into cardiomyocytes so they can study the channel activity in the mitochondria. So this paper that they were working on, once we gave them the lines, these lines did not produce protein in this protein called NDUF-B10. And so what I'd like to point out is this was a very challenging skin fibroblast to work with. The patients were extremely sick. These fibroblasts did not grow very well. We had all sorts of problems. We failed reprogramming several times. Eventually, we started working on the methods and adding a lot of supplements to help these cells proliferate a little bit in order to make stem cells. And it took probably about over a year, maybe a year and a half to successfully make these stem cell lines. But you can see here in panel C, we were ultimately able to make these stem cells. And then Dipayan was able to perform the functional assays and publish this work, which came out last year in Nature Communications. And so that was another example of how our core helped somebody in a pinch to get the work completed. And it was, very exciting to play a part in this study. So another example of work was Dr. Chistani. We collaborate very closely with him because he is intimately involved in making stem cells and doing CRISPR technology. And so we worked on this paper for probably about 7 years, and it got published in 2021. And we learned a lot about how to make stem cells from patients and how to differentiate them into cardiomyocytes. And now we're on to the next phase of creating CRISPRs and differentiating these cells into cardiac organoids. But this paper basically identified a recent atrial fibrillation risk allele that was that appeared relatively recently, about 5,000 years ago in Europe. And then what they did in collaboration with Ancestry.com was they were able to follow this risk allele in Europe, human migration across the US, and ended up in Utah, where this allele is at pretty high prevalence in Utah. And so from this work, we've made, we're working on a couple of different CRISPR projects. And what we're doing is we're taking, they've identified this gene called, it's a transcription factor called NFAT-C1, that is believed to play a role in atrial fibrillation, and also this gene called CD320 that's believed to have a role in left ventricular non-compaction. So we have gotten patient samples and then made CRISPR knockout gene mutations in NFAT-C1 and CD320. This work is in progress, and so hopefully in the next year or so, this can also lead to a publication. We also have several other ongoing CRISPR lines that we've generated for Dr. Dean Tanton, Dr. Nicole Link, and Edwin O, who is out of UNLV in Nevada. And this work is also ongoing and hopefully will lead to future publications as well. So in summary, I kind of described many of the services that our core offers in the area of molecular biology, stem cells, and also biorepository work. We are always looking to collaborate on patient-specific iPSC lines for innovative research, and our lab is fully equipped to make engineered lines for investigators at the University of Utah, but also within the CTSA network. I am thankful that we have CTSA funding in order to push this work forward. And as you can see with the CTRC, over the past 7 or 8 years, we've had many staff and undergraduates that have come through our lab that have contributed to this work, who are now moved on to bigger and better things. Many of them have gone to grad school, gone into biotech, gone to med school. And so we also use our core as a way to train staff for future stem cell research. I'd like to thank the Tristani Lab and Scott Cho, because we have collaborated very closely for the past 8 years plus, working on stem cell development and all the technologies involved in CRISPR. CRISPR work is a huge project. We work with companies as well as 5 other cores on campus, including James Marvin in the flow cytometry core. Brian Daly does all our sequencing, and Crystal Davey makes all of our CRISPR constructs. And so I thank everybody for attending this talk on CTRC's role in trying to develop these technologies for investigators here at the University of Utah and beyond. And I can take any questions about our core if you're interested.
Thank you so much, Colin. Karen Ho, you had several questions along the way. Would you like to first share what institution you're from? I don't know if you're from the University of Utah or elsewhere. I know we distributed this at many different sites. So if you could share where you're from, and then, yeah, I think we'd love to field your questions.
Sure. I have an adjunct position at the medical school at the University of Utah in medical genetics. I'm also affiliated with Clean Nanomedicine, a local pharmaceutical company based here in Salt Lake City. So thanks so much for taking the call. And thank you also for my questions. And thank you also for this great presentation. I run the preclinical studies for this drug company. So that's why I'm interested in iPSCs. I was just wondering if there's a catalog of the 2,500 iPSC lines that you've generated that are available to researchers, and how well they are characterized clinically. For example, do you have information about their disease state, whether or not they were symptomatic or asymptomatic, what those symptoms are, and things like that?
Yeah, so in our core, we catalog these lines, okay, and they're entered into our LIMS system, which is Open Specimen, but we don't keep any patient information in there, okay. That, Open Specimen is searchable, but we try to de-identify these samples, right, when we process them. And so usually if I get an inquiry, the investigator already knows about them, or, I have to relay them to the investigator, for permission and whatnot to use the samples. And that's why the company, Prime Medicine, they needed to sign an MTA for us to ship them the samples. And I think they found out about that we had made those lines through our paper. And then for some reason, they contacted me, which I'm not the senior author, but they contacted me. And then I was able to connect them with Jungyang, right?
Yes.
But I think that, one of the goals of a biorepository is it is a biobank, right? It's like a financial bank. You don't want samples to go in there forever and just sit there statically and nobody ever does anything with them. You want these samples to be used, right? You want samples to go in and you want them to go out for research purposes. And that's why we created them. And as if you, in total, it probably takes like a year to make a line, and then do it, if you're going to do a CRISPR mutation on top of that. Lots of time and money was spent on making these lines, and you want people to use them, right? So I think that at least most of the investigators in our core would be interested in sharing these samples. Whether it's a good point to think about a way that we can maybe advertise the catalog that we have, right? Of all and we have many at this point, as you can tell. We're like a lot of cores. We've spent a lot of time making all these lines, and some of them kind of, they make them, they publish a paper, and then they just sit there, but they're still valuable, right? It's still a valuable research for investigators. So I'm gonna have to think about a better way to have an actual catalog, or at least a list of lines, maybe. And that way anybody can contact me at any time and I can connect them to the PI, right, for permission.
Exactly. Yes, that'd be very helpful. Also, would you characterize your services as kind of stopping at the generation of the iPSC line, at which point you then give it over to the collaborating lab to carry out differentiation to the cell type that they really want? Or do you actually differentiate the cells and then provide them for experimentation?
Yeah, it depends on the lab. So some labs, as you can imagine, are big labs, and they have the staff, or, the postdocs and the graduate students in order to do the differentiation themselves. Marty Tristani is one example. He has, maybe 8 staff that can do that. Another example, we're doing a collaboration with investigator at in Nashville and here at the University of Utah. They don't have that expertise, so we're differentiating them, the cells into endothelial cells for them. And then as I mentioned earlier, we're making some cardiac organoids for investigators. So it really depends on the lab. If they need help, we'll give them help, because we have the experience of transitioning the cells from pluripotent state into the differentiation protocol, and we understand how that works. Sometimes we can train people to do it. Sometimes we start to do the differentiation, and then like a postdoc will learn from us, and then they'll take that work to their lab, and then they'll continue it from there. So we kind of offer a variety, kind of hybrid services, or do the whole thing ourselves. So
Got it. So in relationship to that, then, as you know, then certain cell types and taking them through the differentiation process does not result in 100% mature, for example, neurons
yeah.
of specific cell type that you want in the dish at the very end. Sometimes you have to fact sort them.
Yeah.
Do you have access to a fact sorter?
Well, we're lucky because we're right next door to the flow core. Yeah,
okay.
and so that's probably one of the luckiest things for me because we're flow sorting all the time, and we just get our dish, walk it out, one door down, and we have, so we're lucky that we just happen to be adjacent to the flow core. So yeah.
Great. Thank you very much.
Oh, you're welcome.
I'm not sure if we have other questions, but I just wanted to jump in and add a few things. First, if you're interested in learning more about the prices and specific services, I encourage you to go to the website, and website could be found through Utah CTSI. So there is a link to CTRC core, and there you can find prices and all the information about services. This is number one. And number two, Colin and I offer stem cell course for graduate students and postdocs and trainees once every other year. And we are offering this course this spring. And this is very unique course because it's not only consists of lectures, but it has some labs. And in fact, we have reserved 3 weeks for lab work. And in the past, the students who attended this course, they learned how to work with stem cells, how to reprogram blood cells into stem cells, how to differentiate stem cells into cardiomyocytes and neurons. So it's a great course for those people who are interested in starting with stem cells and learning all the basics related to them.
Any final questions? Well, thank you for joining us today. Colin and Alex, thank you so much for your time and presenting. Hopefully we'll maybe find some new collaborations out of this.